Welcome everyone. Thank you very much for taking the time out of your busy schedule to attend the webinar. My name is Xiaoyu Zhang and I'm working on application and technical support for xCELLigence cardio and CardioECR systems. Today I'm going to talk about the xCELLigence RTCA CardioECR systems for preclinical safety assessment and improvement of iPSC-derived cardiomyocyte functional maturation and assessment of inotropic compounds. First, I would like to give you a very brief introduction of our company, which will be followed by an overview of CardioECR system. In this section, I will talk about the unique features of the system and its major applications in cardio field. At the end, I will show you some CEPA results using CardioECR system and how to use the system to improve functional maturation of human iPSC-derived cardiomyocytes and some results of inotropic compound test in these functionally enhanced cardiomyocytes. Our company, ACEA Biosciences, was founded in 2002. We are the technology inventor of xCELLigence RTCA system. I know some of you have already known us very well, either through our commercialization team or via our distributors. We are now growing very fast. We have more than 500 employees globally. We have already expanded our instruments from one product line to two product lines. NovoCyte is our new brand for flow cytometer. Since it launched in 2014, it has already established very well reputation in the flow cytometry market due to its ease of use software and very competitive features. However, today, I will only focus on our first invention, xCELLigence real-time cell analysis systems. Under xCELLigence brand, we have already developed 7 different models with different throughputs and applications. The major applications for xCELLigence include cell status QC, cell proliferation, cell migration, invasion, and cytotoxicity test. Shifting away from the major application of xCELLigence systems, what I'm going to talk about today is about our cardio and CardioECR system and their application for cardio research. Before getting into the main part of the presentation, I want to emphasize that all of our xCELLigence systems are permanently placed in the incubator to ensure real-time monitor for both short-term and long-term effects of drugs under the optimal cell culture condition. Cardio is our legacy system, which only records impedance signal. However, until now, it's still widely used for the study of general cytotoxicity and contractility of cardiomyocytes. And this is our CardioECR system. From the name you can tell, CardioECR system adds one more readout, ECR readout. ECR stands for extracellular recording, which is used to represent few potential signals from cell culture. Up to now, we have placed more than 80 systems globally and more than 30 publications citing cardio and CardioECR system. The users in pharmaceutical companies use it mainly for cardiac safety assessment. Now we also see the trend that more and more researchers in academic lab or hospitals are utilizing the systems as a reliable tool to study functional phenotypes of disease-relevant cardiomyocytes, which can be potentially used for development of therapeutic strategies. Just in case some of you are not very familiar with our technology, I will talk about how the system detects subtle cellular changes. All of our xCELLigence system use the same technology, impedance-based technology to measure the signal. So this is our plate called E-plate used on the system. As you can see, there are microelectrodes on the bottom of each vial. These small electrodes work as one big positive and one big negative terminals and measure the impedance from the entire vial. So in order to generate impedance First, the system applies a very low voltage, about 10 millivolts, to the plate during each recording. If the plate only contains medium, there will be very low current flowing between electrodes. Once the cells are seated in the plate and start to attach the bottom of each valve, they will act as resistors to impede the current. Therefore, the impedance is affected by cell number: more cells, higher impedance; cell attachment, stronger attachment, larger impedance; and morphology. Different morphologies of cells will generate different impedance values. It has been reported that impedance is 100-fold more sensitive than microscope in terms of detecting morphological changes of cells. So for the beating cardiomyocytes, there will be changes in both attachment and morphology during each contraction of cardiomyocytes. Since impedance can detect cell attachment and morphology changes in a very sensitive way, theoretically, the impedance should be able to pick up contractile signal. But in order to capture these temporal changes during the contraction, the data acquisition rate has to be very fast. Therefore, we increase data acquisition rate of cardio and CardioECR system 1,000-fold faster than our other xCELLigence models. CardioECR system has a data acquisition rate of 2 milliseconds for the entire plate. In order to provide you more direct experience of the system, I'm showing you here is the contraction of cardiomyocytes on our E-plate. This is a 10 days iPSC-derived cardiomyocyte culture. At this stage, you can see the cells have already formed cell monolayer. Due to the physical and electrical connection between each cells, the cells generate synchronized twitching or contraction, if you wish, in the well. This is our data acquisition interface. In order to review the traces on the screen, first you need to select the wells. The traces shown here are the signals collected from the corresponding wells. The color of traces matches the color of wells. Since the CardioECR system records both impedance and field potential signal simultaneously, each well generates two types of waveforms. The top traces are impedance trace, which reflect the physical movement of cell monolayer, and the second traces here are field potential signals. Compared to other assay platforms, cardiomyocyte assays on CardioECR system has more simple assay workflow. So first, thaw the cryopreserved cells accordingly to the cell culture guide from the manufacturer, and then simply dispense the cell suspension into the well of E-plates, just like seeding cells in regular microtiter plates. The cell plates then engage in the CardioECR system. Again, the station is permanently placed in the incubator. The signals of the cell culture can monitor right after cell seeding. Before compound addition, cardiomyocytes need to be cultivated for a certain period of time, which in the range of 4 to 16 days. It allows the cell to form well-connected cell monolayer and generate consistent and robust signals. This pre-IC time varies due to the source of cardiomyocytes. During the cell culture, then you will get this cell growth curve. The cell index is the arbitrary value of impedance. The time course of this cell index is shaped up by the cell index values at each recording times, reflecting the cell growth or cell health. Since iPSC-derived cardiomyocytes do not proliferate, we normally call this curve as cardiomyocyte healthy curve. From this curve, you can see at time 0, where the cell were just seated, and then the cells start to attach and spread out. After a couple of days, it reaches this plateau phase. If the cells are now healthy or dying, you should see a drop in the cell index curve. Besides the cell viability, you can also observe contractile and electric activities of the cells at a millisecond time resolution. What you need to do here is to zoom in each recording time point. In order to get reliable results from cells, it's very important to know if the cells are ready for treatments. The beauty of this system is that you can monitor cells in real time throughout this pre-culture period. Based on the impedance and field potential waveforms, you can evaluate whether the cells are ready for treatments. So if the cell passed the cell static QC for the compound screening, you can start to add compound. The cell responses to the treatment can be recorded right after compound addition. As we know, cardiomyocyte contraction is due to excitation-contraction coupling. This cartoon shows that the signals starts from the ion channel on the membrane, which induce changes in the concentration of intracellular calcium. The calcium will ultimately binds to the troponin C to activate the movement of cytoskeleton. As I mentioned previously, our cardio system measures the downstream event, contraction, in this coupling pathway. Therefore, no matter the changes in contraction come from ion channel modulation, sensitivity to calcium, or interruption on cytoskeleton, it will be all captured by cardio system. However, we didn't stop there. We believe that it would bring more values and mechanistic insights to the researchers if the system can get more information besides cell viability and contraction. Building on the success of the impedance-only cardio system, in 2014, we introduced our second-generation CardioECR system to the market. The ECR system looks very similar to cardio system. However, on the bottom of each valve, there are two separate sets of electrodes, impedance electrodes and two field potential electrodes. Due to the separation of these two types of electrodes, CardioECR is the first platform to allow simultaneous measure cardiomyocyte contraction and field potential signals. As you already know that impedance electrode is used to measure the averaged impedance signals from entire cell monolayer, while these two field potential electrodes are used to measure field potential signals from the cells around these two electrodes. Again, impedance signals allow you to evaluate both contraction and cell viability at different time resolutions. Like other MEA systems, the field potential signal is used to evaluate integrated ion channel activities. Besides providing these two different readouts, the system also provides a built-in pacing function via these impedance electrodes. This is an example of beating waveforms before pacing from 4 different valves. After pacing, the cells in different valves beat at the same frequency and at the same time. Well, there are a lot of in vitro assay platforms out there for cardiac safety evaluation. Then what is unique about the CardioECR system? I will go through these listed unique features one by one in the following slides. First, Laborfree. Laborfree not only helps to save the time used for optimization of labeling conditions, but also avoids the interference of labels with cells. This figure is published in Cardiovascular toxicology. So the data here shows that even the IC buffer seems to be toxic. It stopped the cell contraction 12 hours after adding the buffer. The combination of buffer and dye caused the cell beating cessation at 4 hours. Our cardio data also shows that during the pre-incubation time with the dye and the buffer at the manufacturing concentration, which is 1x, the cell stopped beating at as early as 30 minutes. This data suggests using calcium dye to monitor calcium transient may not be an ideal way to evaluate cell contraction. Second, high predictivity for pro-arrhythmia liability of drugs. We tested a panel of arrhythmogenic drugs. Some of them have been pulled out from the market due to drug-induced arrhythmia. These are traces obtained from before drug addition. The red traces here are impedance traces or contraction traces. The blue traces here are field potential signals. As we can see before compound addition, these traces are very consistent. However, after compound addition, the contraction traces start to show these typical arrhythmic beating patterns, which demonstrated by these small beats in between the major beats. The field potential signals also showed EAD-like or ectopic beats. The combination of arrhythmic beats on impedance traces and arrhythmic signal on field potential traces double ensure the accuracy of pro-arrhythmic effects of these tested compounds. Third, the system provides multi-parametric analysis for cardiac safety evaluation of drugs. As I have mentioned several times, the system evaluates compounds from 3 different aspects: general cytotoxicity, impact on contractility, and ion channel modulation. Bleb setting is our favorite poster child, which demonstrates the importance of checking both electric signal and contraction signal from the same well at the same time. As we know, blebstatin is a myosin heavy chain ATPase inhibitor. It doesn't show significant impacts on field potential signal when you use MEA technology, no impacts on calcium when you use fluorescent staining, and no changes in single channel activity when you use patch clamp. But it shows sensation of cell contraction when you measure impedance traces using CardioECR system. From these impedance traces at different concentrations of blebstatin, we can observe a very good dose dependency. At 10 micromolar, we actually can see blebstatin stop the cell beating altogether, while for the field potential signals, which remain the same. Another example is doxorubicin. Doxorubicin is an oncology drug. On one hand, it kills cancer cells. On the other hand, it has a long-term cytotoxicity to the heart. So from this table, we can detect reduction of the beating rate and the beating amplitude at the late stage of compound addition. If we combine these waveforms to the cell variability curve down here, we can have better understanding on the root cause of cell beating behavior changes. The changes in beating only occurred when the cell index decreased, indicating the cell beating performance changes most likely due to the cell death. The combination of all this information collected from one well provided you very comprehensive assessment for the compounds of your interest. This is another data of doxorubicin, which is generated from pluripotent cardiomyocytes from nCardia. As we can see from this cell variability curve, after 4 days treatment with doxorubicin at 1 micromolar and 3 micromolar, the cell index dropped to 0, suggesting the cell death. In addition, we can see 14 hours post-treatment, we can detect irregular traces from this field potential signal recorded on CardioECR system. Last but not least, the system can be used for the evaluation of long-term effects of compounds. Here, I'm going to use pentamidine as an example. Pentamidine is an ERK channel trafficking blocker. It inhibits the translocation of ERK protein from ER to the membrane, resulting in an indirect blockade of ERK channels. The MOA of pentamidine determines its delayed impacts on the cardiomyocytes. As we can see here, before compound addition, the cells had a very consistent beating activity. After 60 minutes or even 300 minutes, there are no changes. However, if you keep monitoring the cells for longer periods of time, you will start to see the decrease in beating rate and beating amplitude at 900 minutes or 15 hours and afterwards. In addition, we also detected a time-dependent prolongation of field potential duration using CardioECR system as shown in the figures here. In terms of application, cardio and CardioECR systems have been mainly used for prediction of proarrhythmia, detecting effects on contractility, study functional phenotype of disease-relevant cardiomyocytes, and assessment of short-term and long-term effects of oncology drugs. In the interest of saving time, I will give a very brief introduction of those key publications demonstrating each application listed here. First, use the system to predict proarrhythmia of drugs. So this is a paper published by Roche-Nutley in 2013. They developed this arrhythmic risk assessment model using human iPSC-derived cardiomyocytes in conjunction with cardio system. In this study, they tested about 120 compounds. Their results shows the IC model has greater predictivity, demonstrating by this larger than 90% accuracy for QT prediction and larger than 82% for arrhythmia prediction. Second, use the system to evaluate compound effects on contractility. Since I will talk about this paper in my later slides, here I just simply read their conclusion. Impedance-based IC using human iPSC-derived cardiomyocytes provides a predictive model for detecting contractility. Third, use the system to study cardiovascular disease-relevant cardiomyocytes. Due to rapid development of stem cell technology, more and more researchers start to generate disease-relevant cardiomyocytes from iPS cells, and they are searching for reliable IC platforms to examine functional phenotypes of these cardiomyocytes. This is the paper use our cardio system to quantify the changes of disease cells. So the data here clearly shows that the disease-relevant cardiomyocytes have this inherent irregular beating rhythm compared to the cardiomyocytes differentiated from healthy donor. In addition, we can use the system to evaluate both short-term and long-term effects of drug from the same experiment. The finding of this paper suggests that you can have better mechanistic assessment of oncology drugs when you combine the information provided by the cell variability or cell index curve with the contraction traces here. Due to these major applications, CardioECR system has been used for preclinical cardiac safety assessment. Here, I just want to use our CEPA study data as an example. The CEPA project started in 2014. The goal of this project is to develop a more accurate and comprehensive in vitro IC for proarrhythmia prediction. The CEPA project has 3 subgroups. We are part of this cardiomyocyte subteam, which uses human iPSC-derived cardiomyocytes in conjunction with field potential readout to check for missed or anticipated effects from other tests. The parameters required by CEPA are depolarization amplitude for sodium channel activity, firing rate, and the CV of firing period for irregular beating rhythm evaluation, field potential duration for QT prediction, and arrhythmia detection. We have published the CEPA Phase 1 results using iCell cardiomyocytes. You can get detailed information from this paper. Now I just want to show you some CEPA compounds data obtained from other cardiomyocytes. This sequential dosing data were collected from core-free cells. Nifedipine, a calcium channel blocker, it shows a dose-dependent reduction in field potential duration. And GNG303, a slow potassium channel blocker, increased the field potential duration in a dose-dependent manner. And this 431 ERK channel blocker increased the field potential duration and induced arrhythmia at higher doses. The pleurocytes cardiomyocytes from nCardia also show the proper responses to ERK channel blocker, the phytolite. So you can see from this figure, the phytolite induced prolongation of field potential duration and arrhythmia at high concentration. Flecainide is a multi-channel blocker. It decreased amplitude of depolarization spike due to sodium channel inhibition. In the meanwhile, it also increased the field potential duration by blocking ERK channel in pleurocytes. I won't show any data from CEPA phase 2 study since this data will be published soon. Now I'm going to talk about how to use our CardioECR system to improve functional maturation of human iPSC-derived cardiomyocytes. As I have showed previously, the impedance-based contractility assay using human iPSC-derived cardiomyocytes has been well validated and used by several groups. So in this study, they tested 49 compounds in iCell cardiomyocytes. Their data clearly show that this assay provides great sensitivity and good accuracy and specificity. Most importantly, they compared these impedance results obtained from human iPSC-derived cardiomyocytes to Inotropic results, which is generated from adult dog cardiomyocytes. Just a little bit about background on these Inotropic systems. Inotropic is a radio edge detection system. It measures the shortening of cell length or sarcomere from single cell during the contraction. Their conclusion is that the impedance results from iCell cardiomyocytes are comparable to these optical-based contraction data. So this is a typical contraction trace obtained from iCell cardiomyocytes. The beating amplitude is calculated based on the impedance difference between negative peak to the positive peak. We have tested several negative inotropic compounds such as nifedipine and belapstat. Inotropic compounds are those compounds affect force of contraction. The black trace here represent the traces obtained before compound addition, and the right trace here is the trace obtained after compound addition. The results here show that the beating amplitude significantly reduced after this compound addition. Taking all this data together, it suggests that beating amplitude is a surrogate for force of contraction. Even human iPSC-derived cardiomyocytes have been used for cardiac safety assessment, such as CEPA project for evaluation of drug-induced pro-arrhythmia. These cells still show inherent fetal-like phenotype. For example, gene expression level, structure features, and contractile responses to inotropic compounds. So in this paper, they tested several inotropic compounds using Inotropic system. All these positive inotropic compounds displayed either no effects or negative impact on contraction. As we know, the negative force frequency relationship is a hallmark of immaturity of cardiomyocytes. So we are curious about force frequency relationship in the cell model used in our lab. In this study, we used electrical pacing to gradually increase the beating rate. The figure here shows that the beating amplitude decreases while the beating rate increases. This data indicates a negative force frequency relationship in these iPSC-derived cardiomyocytes, suggesting immaturity of these cells. It has been a long-standing challenge to achieve mature iPSC-derived cardiomyocytes. Up to now, several methods have been developed and appear to improve maturation status of iPSC-derived cardiomyocytes, such as long-term cell culture, or use different substrates with different stiffness, or the cell patterning, or provide cells mechanical loading, and also you can add some growth factors to the medium. And among these methods, electrical pacing is one of them. Since CardioECR system can pace the cells through impedance electrodes, we took the advantage of this feature and developed a protocol for improvement of functional maturation of iPSC-derived cardiomyocytes using long-term electrical pacing. So basically, we seeded the cells in CardioECR plate that the cells attach and form a cell monolayer, which takes about a week. Then we will apply a ramp-up pacing protocol to pace the cells for an additional 3 weeks. At the end, we will stop pacing for 7 hours to let cells recover and generate their own spontaneous beating before compound addition. The compound treatment will be tested without pacing. By using long-term pacing protocol, first we want to know if the pacing would be able to reverse the negative force-frequency relationship. In this experiment, we tested force-frequency relationship during this 3 weeks pacing procedure. In order to have a proper control, we only paced half of the plate. The remaining half of the plate was used as non-paced control group. So week 0 here means the cells were cultivated for a week before pacing started. In this control group, non-paced cells, it shows the negative force-frequency relationship on this week 0. Week 1, week 2, week 3, you don't see significant change in this negative relationship. However, when we look at these paced cells, week 0 showed this negative force-frequency relationship. Week 1, week 2, you start to see the significant change in the slope, and week 3, you can see the positive force-frequency relationship. This result suggests that there may be something changed in these paced cells. First, we checked the protein expression level of ryanodine and SERCA, the key calcium handling proteins. However, our Western blot result didn't show profound changes in the expression level. Then we did immunostaining and tried to determine their cellular localization. We actually observed better organization of these key calcium handling proteins after pacing. So in these non-paced cells, we can see the staining for ryanodine and the SERCA are diffused staining. And however, in these paced cells, we actually can see this striated sarcomere-like pattern, which only can be seen in mature cardiomyocytes. This data suggests that paced cells may have better calcium handling mechanism than non-paced cells. So in order to test calcium handling, we treated cells with caffeine, a ryanodine receptor activator, which increases intracellular calcium concentration, resulting in the increase in contractile force. After adding caffeine, we did detect increase in beating rate in both paced and non-paced cardiomyocytes. However, when we're looking at the beating amplitude, we only can see the increase in beating amplitude in paced cells. And this is the trace obtained from paced cells before and after caffeine addition. Then we further tested cell responses to positive inotropic compounds. Isoproterenol is a well-known positive inotropic compound. In addition, it also increases beating rate of heart. So here we added isoproterenol to both paced and non-paced cells. Indeed, we can detect the beating rate increase in both groups. However, for the beating amplitude, we only can see the increase in beating amplitude in these paced cells, and the trace from paced cells before and after isoproterenol addition. So we continued to test up to 6 positive inotropic compounds with different mechanism of action. In addition, we also tested 1 negative inotropic compound, isradipine. All these positive inotropic compounds showed positive impact, and negative inotropic compound has negative effect in beating amplitude. So next, We want to know if long-term pacing can also benefit to the assessment of ion channel modulators. Renolozine is a dual ion channel modulator. It not only blocks ERK channel but also reduces late sodium current. Due to the compensation of blocking these two currents, the clinical data of renolozine doesn't show QT prolongation. But when we look at the data obtained from this non-paced iPSC-derived cardiomyocyte, at this 10 micromolar, the compound increased fuel potential duration and caused arrhythmia. However, in paced cells, we didn't see any changes in fuel potential duration after the compound addition, which is consistent with clinical data. In order to make sure that these paced cells still respond to ERK channel blockers properly, we added the alight after 10 micromolar renolozine treatment. Now we start to see arrhythmia from paced cells. This data suggests that paced cardiomyocytes are still sensitive to ERK channel modulators. In summary, xCELLigence RTCA CardioECR System contains both fuel potential electrodes and impedance electrodes. The system allows simultaneous measurement of ion channel activity, contractility, and cell variability. The system is one of the platforms being evaluated as part of CEPA initiative. The maturation status of iPSC-derived cardiomyocytes can be improved after application of a long-term electrical pacing using CardioECR system. And thank you. Now I'm happy to take questions.